Clostridium, the genus Clostridium contains the most important group of microorganisms that are usually involved in different anaerobic wound infections. These Clostridia are usually gram-positive, anaerobic, and they are specially known for their spores which are spindle, and hence the name Clostridia is given. The genus Clostridia contains three important group of organisms that are involved in the causation of the diseases such as gas gangrene, food poisoning, that is food botulism, and tetanus. For this topic, we'll be dealing in detail about the Clostridium perfringens, that is the organism involved in the most important wound infection, that is resulting in gas gangrene. Coming to the introduction, this organism, Clostridium perfringens, also called as a Clostridium welshi, was cultivated by Achalme in 1891. But the organism was first described by Welsh and Nuttall, who were able to isolate the organism from the blood and the organs of the human dead body, that is a cadaver. This organism is the one that is mostly involved in the most fatal wound infection, that is a gas gangrene. Apart from gas gangrene, it also has the ability to cause food poisoning and necrotic enteritis. Clostridium genus are mostly inhabitants on the human and the animals, and they can be seen as commensals on the skin and mostly shed as contaminant in the feces, and sometimes as a commensal on the human skin of perineum, thighs, and the legs, etc. Coming to the morphology of this organism, these organisms are plump, gram-positive, and they are straight, parallel sides, rounded and truncated, and measuring about 4 to 6 micrometer in length and 1 micrometer in diameter. And they can occur as singly or in chains or in bundles. Most of these species are pleomorphic in nature. They show filamentous type of morphology, and apart from that, involutions forms are usually common in this type of organism. They are capsulated, non-motile, like other Clostridia which are motile by the peritrichate flagella. This organism, which is Clostridium perfringens, is usually a non-motile organism. And coming to the characteristic morphology, that is the appearance of the spores in the organism. The spores of Clostridium perfringens are usually central or subterminal, whereas in the other group of organism you can see a terminal spores. Here in this particular group of organism, that is Clostridium perfringens, you can see the spores that are usually present either in the center or in the subterminal, and you cannot cultivate these sporulating organism on the media. That is, when cultivated on the artificial media, the spores are not appeared properly, and hence the absence of the spores is a characteristic morphology feature of this Clostridium perfringens, and it is easy for the identification in case of cultural characteristic. That is, absence of spores in the artificial media helps in indicating the presence of Clostridium perfringens, whereas other organisms which are sporulating can be easily identified on the artificial media. Now, coming to the cultural characteristics, apart from that, in the introduction part, these sporulating organisms can be easily found in the soil, dust, and airs. Coming to the cultural characteristics, these organisms are anaerobic or microaerophilic. Anaerobic means these organisms cannot survive in the presence of oxygen, whereas microphilic organisms are the one that can survive even in the presence of a little amount of oxygen. Coming to this Clostridium perfringens, this organism can survive in the presence of air, that it is not as fastidious as other anaerobic group of organisms. Coming to the optimal pH at which they satisfactorily grow, it is around 5.5 to 8, and the temperature is 20 to 50 degrees. That is, there is a variation in the different group of organism, and you can isolate this organism in a temperature ranging from 20 to 50 degrees centigrade. But optimum temperature for the growth of this organism is 35 degrees centigrade, and if you want to grow them as a pure culture, the 45 degrees temperature is the perfect temperature at which this organism can grow. And the generation time of this organism is less than 10 minutes. So keeping this generation time and the temperature into the mind, you can inoculate the organism on a specific media, that is Robertson's cooked meat broth media. If inoculated with a mixed culture of different Clostridia and incubated at 45 degrees centigrade for 4 to 6 hours, you can isolate or identify the pure cultures of Clostridium perfringens. That is, you can selectively distinguish this Clostridium perfringens by growing them at 45 degrees centigrade in a Robertson's cooked meat broth, and they are further subcultured in order for to grow them as a pure culture. You can subculture them on a blood agar plates. Now, when grown in a Robertson's cooked media, they usually convert the media into pink, and they do not have the ability to digest the meat. And you can see acid formation and sour odor that is usually coming out of that media, indicating the presence of Clostridium perfringensis. In the lactose milk, you can grow this organism. The lactose is fermented to produce the acid, and the acid that is formed converts the media from the blue to red, indicating the growth of the organism. The acid that is formed usually coagulates the casein that is present in the milk and thereby disrupts it with the production of the vigorous gas. Now, when this gas is produced, it can be easily identified when the paraffin plug is pushed up in a test tube, and shreds of clots of coagulation or the casein is usually seen on the sides of the test tube. So this is called as a stromy fermentation, which is a characteristic feature of the Clostridium perfringensis. So, and hence, in order to identify in the liquid litmus milk the presence of a stromy fermentation indicates the presence of Clostridium perfringensis. Now, coming to the growth of this organism, that is again they have to be subcultured on a blood agar for their pure identification in a pure cultural form. So, in the blood agar, they usually show a target hemolysis, where initially you can see a narrow zone of complete hemolysis due to the production of a theta toxin, followed by a wider zone of incomplete hemolysis, which is due to the production of another toxin, which is the alpha toxin. So, target hemolysis helps to identify Clostridium perfringensis on a blood agar medium. Next, coming to the biochemical reactions, these organism has the ability to ferment many types of carbohydrates such as glucose, fructose, maltose, lactose and sucroses with production of acid as well as gas. In other organisms, we have seen that they either produce acid or gas when they ferment any sugars, but here in the case of this organism, they are able to produce both acid as well as gas in a particular biochemical medium. These organisms are indole negative, MR positive, that is methyl red positive, VP negative. H2S is produced abundantly by this organism, and they are able to reduce the nitrate. So these are the biochemical characteristics that helps in identification of the organism in the biochemical test. Now, coming to the resistance of this organism, because the organism is a sporulating one, it is mostly heat resistant, but the spores can be eliminated by boiling at a particular temperature, and they are sensitive to boiling. But the spores that are produced by the type A and C strains are highly resistant in nature, that is, they cannot be easily killed by boiling, and hence you have to go with the autoclaving at 121 degrees centigrade for 15 minutes at 15 lbs to eliminate this sporulating forms of organism. And these organisms are highly resistant to antiseptics and the disinfectants. So that means you cannot easily kill the sporulating organism by application of antiseptics or the disinfectants. Coming to the classification, how do you classify this organism? The classification of this organism is mostly based upon the toxin that is produced by this organism, and this genus has the ability to elaborate or produce different types of toxins. The toxin that are produced by this organism is more in number when compared to the other group of organism. So based upon the type of toxin that is produced, you can classify the organism into 5 types, and the toxin that are produced on which they are usually classified are called as the major toxins, and typing is usually done with the help of neutralization test with the help of antitoxin. So toxin, in order to type a particular group of organism, the toxin is the most important characteristic feature, and the typing is based upon the neutralizing ability of the antitoxin. So type of antitoxin that is produced against the toxin helps in classification of this particular organism. So there are 5 different group of organism classified from A to E, and type A is the one that is mostly involved in the causation of the gas gangrene. Now, coming to the toxins, which is an important feature of this organism, and most important one, that is the reason for which you can see the pathogenesis of gas gangrene. There are 12 different types of toxins that are produced by these organisms, but the major toxins are usually 4 in number, and they are usually designated as alpha, beta, epsilon and iota. So among these four, as already discussed, type A alpha toxin is the one that is involved in the causation of the gas gangrene. Coming to the alpha toxin in detail, this alpha toxin is produced by all groups of clostridium species. More specifically, it is produced by type A in abundantly. That is, the type A strain of Clostridium perfringens has the ability to produce more amount of alpha toxin, and it is because of this alpha toxin that you can see the biological activity leading to gas gangrene. Apart from this profound toxemia that you can see characteristic of this particular disease, there is also other things that are associated, which is called as a dermonecrotic and hemolytic. That is, the organism not only has the ability to cause gas gangrene but also can cause dermonecrotic as well as hemolytic property. The toxin is specifically a phospholipidase, also called as lecithinase. The lecithinase is an enzyme that helps to convert the lecithin to phosphoryl choline and diglyceride in the presence of calcium and magnesium ions. And the production of this lecithinase can be easily seen when the organism is grown on a media containing egg yolk or the serum, and the production of the lecithin is usually seen in the form of opalescence. That is, the organism which has the ability to produce on a media, that is the egg yolk or the serum, the ability to produce a lecithinase can be easily identified by the opalescence colonies that are seen on the media, and the loss of this opalescence when the media is incorporated with the antitoxins. So this is usually very well demonstrated with the help of a reaction which is called as a Nagler's reaction. That is, the presence of opalescence colonies is demonstrated with the help of Nagler's reaction. The media that is selected to identify the organism that produces the lecithinase or the lecithinase producing organism is it contains 6% agar, 5% Fels peptic digest, and 20% human serum. So here what we do is, in a plate, petri plate, where the media is usually present, we divide the petri plate into two halves. One half of it is inoculated with the antitoxin, whereas the other half of it is devoid of it, after which we streak with the organism and incubate it. Now, the presence of lecithinase can be seen on the other half where there is no antitoxin that has been inoculated or incorporated. So the presence of opalescence around the colony indicates the production of lecithin, which is absent in the area where antitoxin has been incorporated. So this is called as a Nagler's reaction, and this particular reaction is usually used to identify the Clostridium perfringens on a cultural media. Now, apart from 20% human serum, you can replace it with the 5% egg yolk, because it is easy available, and you can the cost of it also comes down when we are using 5% egg yolk. So the opalescence can also be detected on a media which has been substituted with 5% egg yolk. Not only Clostridium perfringens produces lecithinase, apart from which the other organism, the other species are also involved in production of this lecithinase. Example is Clostridium novi, Clostridium bifermentans, etc. And this organism not only produces opalescence but also have the ability to disrupt the erythrocytes. That is, they can cause the destruction of the RBCs by targeting certain phospholipid components on the RBCs, and hence they can be seen on a blood agar media by a pattern of hemolysis. So hemolytic property of this organism is because of the action on the phospholipids by the lecithinase, and this can be very well understood with the help of a phenomena which is called as a hot-cold variety. And the organism is inoculated on a blood agar, incubated at 37 degrees centigrade, immediately after which it is chilled at 4 degrees centigrade. You can see the disruption of the hemolysis or the hot-cold variety phenomena. And the toxin is highly heat stable in nature and can be inactivated, or you can say it is partially inactivated while boiling. Now coming to the other toxins, that is beta, epsilon, iota. So these are also the toxins that are involved in the pathogenesis but not as lethal as alpha toxin. They have a necrotizing property, and gamma and eta are the one which are less lethal when compared to the other type of toxins. Coming to the delta, it is lethal and both hemolytic in nature, and theta is cytolytic and it is oxygen labile. It is similar to that of streptolysin O of the the Streptococcus species. And kappa toxin is the one that produces collagenase. This collagenase helps in the spread of the infection. And lambda toxin is the one that elaborates or has the property of proteinase and gelatinase. And mu has the property of hyaluronidase. Hyaluronidase is an enzyme that breaks down the cementing material between the cells, that is the intercellular cementing material that is present can be broken down by the production of this hyaluronidase enzyme by the mu. And Nu, that is the one that helps in elaborating deoxyribonucleases that targets the DNA. Apart from these toxins, the organism also has the ability to release certain soluble substances which are enzymatic in nature, and these enzymes have the ability to act on blood group substances and A and H substances, and they are neuraminidase that goes and interact with the myxovirus receptors of the RBCs, and they release certain substances. These enzymes release certain substances that renders the RBCs pan-agglutinable. Apart from that, they also show hemagglutinin activity. They also have a fibrinolysin activity, that is the enzymes are fibrinolysin in nature, and certain bursting factors that you can see in this particular disease, that is the spread of the infection is because of the formation of certain gases that are involved in the spread of the infection. So they are called as the bursting factors, and these bursting factors can be seen in the characteristic lesion that is produced by this organism, and they produce circulating factors which makes adrenaline sensitivity in the capillary bed and also inhibit the phagocytosis. So the organism not only is able to produce different types of toxins that is involved in the pathogenesis of the infection, but also releases certain enzymes that helps in the spread of the infection from one part of the body to the other. Now coming to the disease, that is a gas gangrene, which is a fatal wound infection caused by Clostridium perfringensis. As already discussed, type A strain has the ability to produce or give a profound toxemia when allowed to multiply in a favorable conditions. So the organism does not cause infection singly, but in combination with the other organism. So it is Clostridium perfringensis along with certain other Clostridium species and non-Clostridia anaerobes that is combined or they show combined effect in production of this gas gangrene. And most of these infection usually is limited only to the surface area, that is it either gets limited up to wound contamination or anaerobic cellulitis. And if the organism has the ability to enter into the muscle tissues, they are able to give rise to gas gangrene. So before passing on to the muscle tissue, the wound infection gets limited only up to anaerobic cellulitis, and if the organism has the ability to invade into the muscle tissues and multiply in the muscle tissues, it has the ability to produce the toxin and cause the gas gangrene. So you should make it clear in your mind that if the organism is only moving up to facial planes, it will not lead to gas gangrene, but once if the organism has the ability to enter into the muscle tissues and multiply in the muscle tissue, then it will lead to gas gangrene. Otherwise it gets limited only up to anaerobic cellulitis. The description of the gas gangrene was first given by Oakley in 1954, who defined gas gangrene as rapidly spreading edematous myonecrosis, which is characterized with a muscle lesion that is contaminated mostly by the Clostridium, especially the Clostridium perfringensis. And it is also called as a malignant edema. Apart from that, the other terms that can be used for gas gangrene is anaerobic myostatis and Clostridial myonecrosis. And this disease was thought to be a disease of the war, that is when the person get wounded due to the bullets and the shells remain inside the muscles. These usually lands up to gas gangrene. But apart from that, the civilian life also are affected by the gas gangrene due to the road accidents or the crush in the muscle tissues due to certain road accidents or any other wounds that are usually caused to the person can lead to gas gangrene. And rarely it usually is affected, or the organism enters by the surgical operation. Mostly during the amputation part, the organism will have the ability to invade into the muscle tissue and cause the gas gangrene, but it is a rare phenomena when compared to the other, that is in the battlefield or the road accidents. And here when you see the organisms that are associated with the gas gangrene, the bacteriology usually varies. As already discussed, it is not only one organism that is causing the gas gangrene, but is a mixture of organisms that is combined giving the effect. So here we will see the organisms that are involved in detail. First, the organism responsible is the clostridia, along with the anaerobic streptococci and facultative anaerobes such as E. coli, proteus and staphylococci. So as discussed, clostridia, the most common type of organism, that is 60% of the organism that contributes to gas gangrene is by the Clostridium perfringensis. Other organism associated with this is Clostridium novi and Clostridium biformentis, which contributes only about 20 to 40%. And apart from these three organisms, the other organisms that are associated are Clostridium sporogenes, Clostridium phallax, Clostridium biformentis, Clostridium sordi and Clostridium tertium. Now, the organism Clostridia has the ability to enter into the muscle tissue only after there is a damage to certain parts of the body or when there is a wound. And as this organism is a normal inhabitant on the skin, it can enter into the muscles and cause the infection. So, this type of infections are usually called as the endogenous infections. So, rarely by the surgical procedure, and sometimes injections such as adrenaline can also cause gas gangrene, because they create an environment for the growth of the organism in the muscle tissue. And based upon the type of wound that is seen on the person, you can McLean classified the wounds, anaerobic wounds, into three types. One is surface contaminant or a simple wound contamination, where you can see the presence of the organism in the wound, but there is no invasion to the underlying tissues. You can say it as only a surface wound contamination. Next is, if the organism has the ability to enter into the facial planes, the organism has the ability to produce a toxin in a limited amount, not as we see in a profound toxemia in the gas gangrene. So in the facial planes, when the organism gains an entry, it multiplies and produces toxin to a less amount. Again, here there is no involvement of the underlying tissue. It is only when the organism has the ability to enter into the underlying tissue or the healthy muscle tissue, the organism is able to cause the most severe infection, that is a gas gangrene. So first is a simple wound contamination, where you can see the presence of the organism only on the surface. Next is the anaerobic cellulitis, where the facial planes are involved but not the underlying tissue. And here when the organism is able to multiply in the anaerobic cellulitis, the disease can range from minor gas abscesses to the complete involvement of the limb. So here a little amount of gas can be produced, or if it is ranging from a little gas, it can also lead up to the involvement of the limb, but not that fatal as we see in the gas gangrene. And the third one is anaerobic myostatis, also called as the gas gangrene, which is most fatal when the organism gains an entry into the muscle tissue. So here the organism, once there is a wound, it will have the ability to penetrate inside the healthy tissue and multiply in the healthy tissue. Now once the organism has the ability to multiply, it will release the toxin, which will show its pathogenic effect. So it is not that the organism can directly produce a toxin. It needs certain favorable condition that allows the organism to produce the toxin or elaborate the toxin. And the most important condition that is required for the organism to multiply and produce the toxin is a low oxygen tension. Now when this could be attained, it is mostly seen in the battle wounds, as already discussed. When the person is hit with a bullet and the shell remains inside his arms or legs, the organism starts multiplying. There is a decrease in the oxygen tension, and as a result of which the organism gains an entry and will have the ability to multiply. Now the necrosis that is seen in this condition is because of the ionized calcium and the silicic acid that is present in the soil. Apart from the battle wounds, the other reasons that usually decreases the oxygen tension or creates a low oxygen tension is crushing of the tissues or the tearing of the arteries. And extravasation of the blood, that is the leakage of the blood, increases the pressure on the capillaries, thereby decreasing the flow of blood or the supply of the blood to the wounded area. As a result of which, again, when there is low oxygen supply, there is anaerobic condition in that particular area, thereby decreasing the oxygen tension. So the EH and the pH, that is the reduction potential, and the pH usually comes down when there is no blood supply to that particular area. Not only this EH and pH, but also there are certain other chemical reaction that takes place in the wounded infection, along with the carbohydrate fermentations and the release of amino acids from the protein. All these together creates a satisfactory environment for the growth of the organism. Now when the organism starts multiplying, definitely it will produce the toxin. So it proliferates and elaborates the toxin that shows its pathogenic effect. And as already discussed, now when there is a extravasated hemoglobin and myohemoglobin, they will cease to supply the oxygen to that particular area. Now when there is a cease of the supplying of the oxygen to that particular area, there is again a decrease in the oxygen tension, thereby making the environment satisfactory for the growth of clostridium species. So extravasated hemoglobin and myohemoglobin ceases the supply of oxygen to the wounded area, shifting from the aerobic oxidation to anaerobic respiration of the pyruvate, thereby producing different types of acid and the enzyme which are followed by the spread of the infection from one part of the body to the other part. And here when the clostridium multiply, the organism releases the toxin. As already discussed, the toxin is of alpha type, that is the lecithinase are usually produced. The lecithinase damages the cell membranes and increases the capillary permeability, because of which more pressure is applied on the capillaries and less blood is supplied to that particular area, and the extravasation and the increased tension in that area again decreases or makes the condition anoxic in nature. Now during this particular thing that is going on in the wound, you can also see the RBC getting targeted with the help of lecithinase. So lecithinase not only produces the anoxic condition in that particular area, but also will destroy the RBCs by acting upon the phospholipids of the erythrocyte, and hence you can see a hemolytic anemia and a hemoglobinuria. So not only it is affecting the wound, but also it is destroying the RBCs. Now when there are less number of RBCs, there is no oxygen that can be supplied to that particular area. So lysis of the RBCs is because of the alpha toxins. Now when this wound infection is going on and the activity of the lecithinase is going on, as already discussed, the organism is not only able to produce the toxin, but also releases certain types of enzyme that helps in spread of the infection. So the enzyme that is produced is a collagenase that breaks down the collagen barrier present between the two cells, and hyaluronidase, which acts upon the cementing material between the two cells, break down the cementing material and will have the ability to get spread from one part of the body to the other part. Apart from this, a gas is usually produced. Now when this gas is produced, it usually decreases the blood supply by an effect which is called as a pressure effect. Now this gas is so strong that it is able to break down the barriers between the two cells, and hence there is a progressive disease that can be seen from one part of the body to the other part. So the disease that is spread is not only because of the toxin, but also because of the enzyme that are produced by this organism. Coming to the incubation period, it is as short as 7 hours to about 6 weeks, and different organism shows different types of incubation period. Clostridium perfringensis usually have 10 to 48 hours of incubation. Clostridium septicum usually has 2 to 3 days, and Clostridium novis has about 5 to 6 days of incubation period. And the symptoms that are associated with this is pain, tenderness and edema in case of localized wound infection, and in case of systemic you can see the toxemia, that is the presence of toxin in the blood. Now when this when the toxin has the ability to enter into the blood, it can cause the destruction of different tissues that are present in the body, that is the spread of the infection. So the characteristic feature of this disease is the release of a thin watery discharge, which later on gets profused and seraguinous, consisting of the blood and the other fluid. And the presence of the gas in the tissue makes the tissue crepitant, that is making a crackling type of sound that again is helpful in the spread of the infection. In most of the untreated cases, it can lead to death of the person due to the circulatory failure, and in these persons you can see there is a profound toxemia that is seen in the blood of the infected individual, along with the prostration which develops due to the toxin that has been spreading throughout the whole body. Coming to the lab diagnosis, this organism can be easily identified based upon the gram staining and the isolation of the organism in the pure culture. But before going with this identification, you have to distinguish between the different types of anaerobic organisms that are involved in the wound infection. So the lab diagnosis should be completely based upon the clinical backgrounds, and the lab diagnostic person should be able to identify the organism based upon not only the identification but also the enumeration of this organism. So the bacterial examination helps in differentiating between the anaerobic streptococci myostatis, which is involved in the wound infection, and the gas gangrene. So gram staining, based upon the gram staining, you can identify the organism as streptococci or the bacilli. In case of streptococci, as we already know, this organism appears spherical in nature and they are arranged in chains, that is a streptococci. This is the initial identification of the organism. Now coming to the clostridium perfringensis, these are the organisms which are bacillus in nature. So based upon the morphological characteristic, you will be able to distinguish between the streptococci and the clostridium perfringensis. Next is the isolation of the organism in pure culture in different media. For that, the specimen that is mostly used are the films from the muscles which have been infected or the wounded. The specimen is usually taken from the edges of the wounds. Next sample can be tissues from the necrotic area or exudate from the deep wounds. So it is in the deep wounds that you can see the activity of the organism, and from the deep wound it is easy to isolate or collect the organism with the help of capillary pipette or the swab. So in the deep wounds, for the collection you can go for the capillary pipettes or the swabs. Apart from that, the other samples can be the necrotic tissue or the muscle fragments. So once the exudate has been collected, you can go for the gram staining, which gives us only a presumptive identification of the organism, because there are different types of clostridium species that are involved in the gas gangrene. One should have a knowledge which species is usually causing the disease, and the different species that are involved in the gas gangrene, the most common one is clostridium perfringensis. This can be identified with the help of gram staining. The characteristic feature of the clostridium perfringensis is that they are large in number and they are regular, and they are usually gram positive bacillate, and the absence of spores is a characteristic feature of the clostridium perfringensis. Next is clostridium septicum. On the gram staining, morphology can be identified as they show citron bodies, leaf or boat shaped structures, and they stain irregularly, and they are pleomorphic in nature. Next is clostridium novi. Again, the organism which is involved in gas gangrene can be identified in the gram staining based upon the features, that is they show large bacillus, they are oval, and subterminal spores are usually seen. So in clostridium perfringensis, absence of the spores is indicative of the presence of the organism, and in case of novi, the subterminal spores can be seen in the gram staining, and they can be identified as clostridium novi. And clostridium tetani or clostridium tetanomorphum can be identified as they are slender bacilli, and they are round, and you can see the terminal spores. So all these four are involved in gas gangrene. The most common one is a clostridium perfringensis. So one should have a detailed knowledge about which type of species is causing the disease, so that the treatment can proceed. Next is growth of the organism in the media. So because the wound infection can be caused by both aerobic as well as anaerobic group of organism, both type of cultures has to be inoculated, and in the case of anaerobic culture, anaerobic cultural techniques has to be used, and the media that is used is a heated blood agar, and you have to incorporate about 5 to 6% of the agar in order to prevent the swarming of the organism. So clostridium perfringensis has the ability to swarm in a media media containing less amount of agar. So you have to increase the concentration of agar to 5 to 6% to prevent the swarming. And identification is based upon the Naglers reaction, as already discussed in the cultural characteristic. Naglers reaction helps in identification of lecithinase producing the organism in the form of opalescence. So incorporating antitoxin can inhibit the production of the opalescence colonies. So that is a Naglers reaction, one of the characteristic feature that helps in identification of the organism. Next is you can inoculate the organism on a Robertson's cooked meat broth. 4 test tubes are usually used and inoculated with the organisms, and each of the test tube is heated to 100 degrees centigrade. First test tube for 5 minutes, second for 10 minutes, third for 15 minutes, fourth for 20 minutes. So with the help of this, you will be able to identify the organism which produces heat resistant spores or the sporulating organism. Now further confirmation is done with the help of blood agar, that is subculturing on the blood agar plate. And in the blood agar, the hemolysis gives a positive test, and most of the clostridium perfringens usually shows bacteremia. That is, without the production of the gas gangrene, this organism can be seen as a contaminant in the blood, that is a bacteremia, and hence this organism can be identified and distinguished from the one that is able to cause the gas gangrene by morphological, cultural, biochemical and toxigenic substances. So lab diagnosis completely based upon the gram nature and the isolation of the organism in pure culture. Coming to the prophylaxis and the therapy. Prophylaxis control, the surgery is the only prophylactic and therapeutic treatment for this disease. That is, you have to completely eliminate or remove or excise that part where which has been contaminated with the organism, and the damaged tissues have to be removed and the wound had to be eliminated with the bits or the foreign bodies that have been implanted during the wound infection. That is, either it is a dust or it is a soil or the cloth, or you can say the bullet that has been wounded to the soldier. So removing of the damaged tissue is only the way to prevent the spread of the infection. And uncompromising excision, when there is an established infection with this organism, uncompromising excision is the only treatment you can say to prevent the spread of the infection. That is, you cannot revert it back to the normal one, but you will prevent the infection to spread from one part of the body to the other part. And if there is an availability of hyperbaric oxygen, that will enhance the treatment procedure. Hyperbaric is supplying more amount of oxygen to that area to prevent the growth of the clostridium, because it is anaerobic in nature and it requires low oxygen tension for the growth and multiplication. So in order to prevent the growth and multiplication, more amount of oxygen has to be supplied to that particular area. That is called as hyperbaric oxygen therapy. Now this treatment can be done in combination along with the antibiotics and the surgical methods. That is, the two-way method to treat this particular infection is use of antibiotics and the surgical method. And the antibiotic of choice that is used is metronidazole, that is given 4 hours before the surgery, followed by 8 hours for 28 hours. And as this wound is usually caused by mixed group of organism, then you cannot depend upon narrow group of antibiotics. So you have to select for broad spectrum of antibiotics that will be able to eliminate both aerobic as well as anaerobic group of organism. And the antibiotics are usually given in combination, where we use metronidazole. Next is we can go with the gentamicin and amoxicillin. So all these three are given as broad spectrum antibiotics to prevent the spread of the infection and eliminate the infections. Apart from that, you can go with passive immunization, that is the anti-serum for gas gangrene, that is the anti-gas gangrene serum can be used in the cases where the person is not able to produce the antibodies in response towards this particular infection. So directly you can go for passive immunization. And this is all about the clostridium perfringens that is involved in gas gangrene.